Okay, shall we make a start then?
Yeah.
Right.
Okay. Well, welcome everybody to our virtual app Roadshow. Steve Donovan is going to kick off first and talk about the development of the app. It's not completed yet, but we want to show you the screens, and we'd like your feedback on the screens as well. And we're going to have a Q&A after Steve's presentation. So if you can put your questions in the chat box, and if I can ask you to do that after Moyne's spoken as well, and then we'll have another Q&A at the end. So everybody could also keep their sound on mute as well, because we are going to record this evening's event. Thank you very much. So would you like to begin, Steve?
Yeah, sure, I will do. Been given 15 minutes, so I'll get my timer going. I didn't work out how long for each screen. I'll just wing it. I'm okay winging it. That's
fine. Don't worry.
Right.
If we overrun a little bit, don't worry.
All right, cool. Hello, everyone. As Wendy said, I'm with the Chief Operating Officer, T-Boy, Sweeper Upper from Citrus Suite, and we're developing the app, which I'm going to take people through now. So what we've gone and done, we're gone and presenting it in. So we did a period of design. We were going back and forth designs, and then once we got had a really great brief from Wendy and the team, and Fiona, and we from that brief we did the design doc, and then we did some wireframes, and then for wireframes we started doing the Envision. Which is an Envision is what we call a posh wireframe, basically, and it kind of shows the design and the user experience. And what I say by user experience, it's like, well, what does this button do once you press it, and what information comes up, etc. All right, hang on, let me see, this puppy wants to come back in 1 second. Yes, he does, just all the time. Go on. Every time I do anything I'm saying, he just pops in and out. Right, so what I'll do, without further ado, I'll show people, so we're presenting it in what we call an Envision, which is basically just a wireframing tool that we use here at Citrus Suite. So what I'm going to do, I'm going to share my screen now and take people through the screens and the user experiences, okay? So this is what we call an early prototype as well. So we still may change once we get feedback and stuff like that, etc. Okay, so what I'm going to do is going to share my screen. Right, and can everybody see that? Thumbs up, yeah, okay. Right, here we are. This is the home screen. This is the first screen you see when you open the app. So we haven't really got the names or anything in there at the minute, what it's going to be called. There's some ideas that have been flying around and stuff. Wendy said some and everybody. So we are going to finalize on a name for the app and put it in stock and get with the branding. But as you see so far, it's kind of looking nice. We wanted a nice soft touch and feel to it, just so when you open it up, it doesn't look like you generally like a medical based app, right? So what we got here, we got what we call the home screen. And we've got certain features that are on the home screen because this is the place where you want to look at for your quick snapshot overlook of everything, right? So I'll just start going down the screens. So you're going to see your name, you're going to have a profile picture and stuff. So when you go into the profile, so we did one of the patient design groups that we did when there was you put your more information in that they want that was needed. So you just got your name, etc., your date of birth, your age and stuff. But we also wanted to know a little bit about you and about yourself, really, and anything you want to put in this bit of information here about me and stuff, just so it looks because the app is got to be part of a researcher's tool as well. So basically, it gives the researchers a chance to know a little bit more about the users. Really, if you've got children and other things that are going on with there, going on like that, etc. Right, and in here you're going to have a chance to put in your immunizations that are outside of the condition, and then what additional drugs you may be on that you're on outside of the condition that you got. Right, so that's that. So that's like the profile screen. As you got going down now, On the left, you can see it's a quick snapshot that says if you're in remission or you're in relapse. I'll go into that a bit more later on. But what we've got there, we've got like a quick snapshot of the date. So you may be like 5 days in remission from this date, and the last time you're in relapse was 30 days ago, etc. So it's a little snapshot of logic, a little snapshot of an overview, what you can see there. And, you know, we're going to the logic of that a bit more. So who says I'm in relapse even though I've got a +3 or +4, you know? So we've got that logic coming in. So I'll explain that in a minute. So then we've got the UPD, your last readings on the right. Again, that's just your urine dipstick stuff. And again, it says if you're in relapse or not and the date it was taken. And also if you've got a pop-up alert, because the app kind of gives you little bits of nudges to say you're in, you know, you've got a high reading and you want to go see someone to get there and see them and stuff. So that's all that's your basic stuff. But then we wanted to make the app a bit more than just medical information and information about your condition, like the hard facts of your condition. So we put a section called Did You Know? Now this bit here, the Did You Know bit's going to be, it might be just little bits of snippets of information that the team want you to know. There may be some new medication, maybe some new treatments, or there may be an event coming up. And with this, you get the chance to see what the event is. And so again, I'll go into that section in a bit. And at the bottom is like your appointments. So you might have any appointment you want to put in into the app. It doesn't give you an alert when the appointment is up and coming, but what it does, it gets you like, there you got to go get a check. But then it gives you the chance to kind of write your note, like I suppose your treatment plan in there when you speak to your clinician and stuff like that. And you can add an appointment. Then you got appointments, graphs, readings, and Did You Know bit there. So what I'll do, I'll start with the readings, right, which will go into the relapse remission logic. There, okay. So what we've got there, I'll just add a reading. So it's going to automatically fill in the date of the reading, and then it gives you an option to kind of put your reading in from a drop down, and it's going to go from negative trace to +1, +2, +3, +4. Then you can put your blood pressure in, you can put your weight in at the time and your body temperature, and if you have any edemas, swollen ankles, anything like that, really. So, but when we did the patient group meeting as well, what came out is like some people may have like a standard reading of +4 and +3. So originally the logic was going to be if you have +3, like +3, +4 readings for 3 or 4 consecutive days, then you're automatically going to get like a relapse notification. But again, some people say, well, that doesn't work for me because I'm kind of +3, +4 most of the time anyway. And so therefore, so what we did, we looked at it again, and we're giving people the choice to go whether they choose whether they're in relapse or remission, and that will set the alert notice up for people as well. Okay, so you can do all that basically. So even in relapse, you can add the medication for where you're in relapse, and it'll attach a tag. Why do you think you're in relapse? And there's going to be a drop down of that type of stuff going on there. Right, so when you submit, I won't do that submit bit yet because that's something else. So if you put in +4 or +3 reading and the tag of relapse, then once you submit it, like if you do it like say if it's your 4th or 3rd time and the tag is in if it's your 3rd consecutive day of taking a reading and you're putting a tag of say you're in remission, sorry, you're in relapse, then you do get that pop up, yeah, with UPD +3 +4, please consult your doctor. It's just little reminders, you know, that you can be able to turn them off as well. Okay. So let me go back to there. So that's in the view readings bit. So in this as well, we've got, so you're going to be you're basically going to be doing like a lot of readings and stuff. So you can filter it as well. So I want to see all the relapse reading or all the remission readings basically. So it's where you can filter all this information. Then you can put in what medication you're on, and when you're in, and if you're being hospitalized because of it. And you can put more information in about your discharge date, your admission date, etc. All right. Not going too quick, am I? Is everyone okay so far? Yeah, cool. I'll take this. I'll take the thumbs up as a okay then. Right. So let's go back to here. Right. Actually, go back to that reading bit. A few readings there. So medication, yeah, you can add your medication, as I said, from drop downs. So these are going to be auto-fill stuff as well. So we're going to get a list of stuff from the team that you can put in. So you're not going to be having to write the medication and all that other stuff in. It'll be like a drop down stuff that's going on there. Right, and again with hospitalization as well, you can add an admission, like the date you were admitted, the discharge, and your reason for admission. Okay. That's cool. So that's like the kind of nitty gritty of it, of where it is. But then I'll just go through a lot of this other stuff now. Then I want to kind of swing back to the graph stuff. So let me kind of do that now. So we've got your appointments. Yeah. So if you had an appointment, again, you can put your time, your date, the appointment, the comments, and then there's be a treatment plan. I say someone did say, well, can there be an alert? But it's just a visual reminder of my appointments. You can look at all your appointments, but it's the treatment plan really where you put down the notes of stuff what you want to discuss with your clinician. So that was your appointments, right? So let's kind of go to did you know? I say with the did you know section again, we're given the ability to add just different types of media. Really for the team that they may find people may be of interest really. So you can add a link to your website. There'll be links to like just bits of information, but you can also put videos in it too, videos, PDFs, any kind of bits of content that you want really. And this will be available and updated as often as the team wants it. As well. So let's go into fluid intake. As you see there, you can get a video, and it doesn't have to be one bit of content either. It can be like lots of little bits of content. But then we're creating a portal where we can flag stuff to be sent to the main page, to the home screen of it. Right. So that's that, and then we've got I say your readings where you can filter them, which I've already shown you. And then you've got the graph stuff. So we kind of went back and forth with Fiona on this a little bit, didn't we, Fiona? Because it was like we had the graph idea, but Fiona was like, well, we really want people to see more, really, and it's a good idea to put different types of medication on it too. So if you have a reading, when you have a reading in line, you can kind of see, you can see what medication you're on that day and what your other things were, like your weight. But we can also so just dead briefly at the top, we could change it to a weekly view, a monthly view, yeah. But then we've got this here. So if you see on the right, it says weight at the minute. If I was to click blood pressure, then that'll turn the blood pressure, and you'll have the blood pressure reading coming up. So also, what we didn't want to do as well was fill that little space up on the mobile phone with full of dots. Yeah. So if you do 2 or 3 readings in a day, really, you don't want to kind of have to scroll in and look. So what we did, if you click that, then we'll see that main reading for the day. But if there's 3 readings done that day, you'll be able to scroll through the readings, and that gives you all the information for you want, which there. So it gives you the your weight, blood pressure, what the reading was, what the tag was, and what the medication is that you're on, really, as well. So that's that. But we're also going to have the ability in it to export the data as well. So if you want to go to your clinician with this, you can export this data and export the pages, really, or the information, so you can see, and you'll be able to email it to your clinician, or you'll be able to print it off as well via Bluetooth and stuff as well. So that's that. And I've got 1 minute 50 seconds left. I think that's it really on it. I say it's still like I think I've shown this to the team and everyone's really happy with it. So we've started actually deving it at the minute, and we're running with deving it concurrently with the portal stuff as well. See, the portal is where all the information is going to be going back to. Just a little bit of tech about the portal is that it's going to link up to the databases, Radar and the UK Renal databases as well. So this information goes up. So you're part of the app, really. You're going to consent to letting the researchers look at information and sending that information back and forth to the other databases too. From that. So you know, so I think that's kind of that's me done. I've got a minute left. Anything I can fill it with for a minute? No, you're all right. I think that's it. So yes, so that's where the production is at the minute. All right. Cool.
That's lovely, Steve. Thank you very much. Thank you.
Right. It wasn't too quick, was it? Everyone understood that, didn't they?
Yes. Well, I could follow it. If there was is there any questions at all? I haven't seen anything in the chat box so far. I mean, does anybody want to ask any questions?
I mean, it looks really nice, Steve. Well done. I've chatted to you before, and I think it looks really user-friendly and has got most of the features that we talked about. Obviously, we had a bit of a discussion about the relapse, but I think the way you've handled it is fine. I'd be interested to see how Martin thinks about that as well, because obviously there's two groups of patients, some who relapse and go into remission, some who just have persistent proteinuria. So you need to sort of cope with both those scenarios.
Yeah, I think with that point, I have put a note in the documentation really for that as well, because we want an additional tag, don't we? I think we said, didn't we? So what we'll do, I'll hook up with you anyway to discuss that some more about what we can do, because I say it's still kind of, you guys, how we generally work is like work with partners like yourself that have all the information. So if something doesn't work or it's not, it kind of does, but it needs a little bit of refining. That's where we'll hook up again, actually, and go through it. But yeah, that's fine.
Yeah, it would be interesting to hear from sort of patients who haven't seen it so far, what their initial impression is.
Yeah. I think probably we'll have to have another virtual event to show the whole thing in motion. But obviously it's got to be synced into Radar first, go through all its certification, and also we've got to test it thoroughly, haven't we? So there is one. Oh, there's a couple of questions in the chat box now, Steve. So the first one from Tina is, she says, great app. Can we select what information we can share, please?
Yes, we can. So there's going to be, you can filter the information out. There's going to be lots of filters, what we call a filter modal. So when you're actually, it's not going to be a blanket export. So there will be stuff in there you can go, oh, yeah, I'll export this and export that. So you don't have to do it all at once. And there's also going to be date filter too. So you can export from either one day or a certain amount of days over the period.
And the next question is, there are two different medications specifically for relapse and routine.
That's me and Wendy, really, just to make the point that you can, patients can record their routine meds, and specifically we're asking them to record their meds that are specifically for the relapse, and that will be listed. But there is a place where they can record their routine ones, because we felt that they would like to be able to do that. Something that came up in the last meeting. That's right. Thank you. Because sometimes the hospital doesn't always prescribe them. They get some from the GP and some from hospital. So I think it was a good place where they could record all their meds. But then the special ones for the relapses would be a separate place.
Yes.
Thank you. Whilst you're on at the moment, Fiona, obviously, to download the app, you need to be really registered with the Renal Registry.
Radar. Yeah.
Radar, sorry, yes.
What we're going to be asking is people to go to their nurses or consultants and ask to join Radar, because it's the only way we can actually join you up. We have to have a consent form signed. So, but that can be done online. We can send an e-consent form out, but we have to be recruited through their own clinicians.
And we're also trying to work out a way so that the hospitals that are currently not signed up to the Renal Registry can actually download the app as well, aren't we? So we're looking into that.
Yeah, and I think that's something that we've discussed, haven't we, Fiona? We were talking about, and Wendy, we were talking about, like, we want the app freely available, but when they can download it, there's going to be, you know, some key information that needs to go in. And if you haven't got all that key information, then it'll be sent to some admin that'll then kind of direct that user to register, you know, and then they'll be able to get on it. Because what we don't want is, like, you know, a lot of people downloading it and they got nothing to do with the renal log or anything, you know, really. So we have discussed that, and that is one thing that we're still tweaking it. Because I had a meeting the other day, I'm like, what information are we going to, because we've got to verify against information as well, and it's a verification registration, which makes or breaks a lot of apps, believe it or not, you know, because the registration process has got to be really easy, but we have to kind of verify against details, and we think about verifying against medical ID and then a date of birth, but we need a third bit of information, and it suggested, can we have a name, but you can't. But names are always like for example, my birth name, my mother always calls me Stephen, but I put me down as Steve's everywhere. And so unless it's a perfect match, when you talk about this type of information, you can't have like matches, you need perfect matches. So we're still refining stuff like that as well, and how we're going to make it a seamless user journey for when people want to register, and how can we connect them to the right renal unit to register as well.
Yeah, so that's going to take a little bit more working out, isn't it? Our next question is, do we have a launch date? Well, we haven't quite yet, have we? You said something about June, July, didn't you?
I think we're looking at July, but all depends what the IG is going to be like, really, on it. We're getting to have, so the way the processes are, we generally have like an alpha, then a pre-beta, then a beta, then a master gold. And so what we'll do when we get to like a pre-beta, we'll probably send it out to users for some user testing and stuff. And so again, get in touch with Wendy and Sandy about if you want to if you want to do user testing and stuff on it. So that's the process. But we are looking toward the end of July, really, for the proper launch. You know, I say a lot of hard work's been done, to be honest, with all the initial designs and the meetings and stuff. So it's just putting it together and working on the little points, you know, the little bits of logic that Moyne's mentioned, and the little bits of like the registration and stuff, which Fiona and what's her name brought up as well. So there's still stuff to do, but we are getting towards July.
Wonderful, thank you. Will it be linked to patient view?
I don't think so.
No, I think I don't think it will either. So
I don't think so, but I say
we might be able to put a link to PKB in there from the NHS app. There's a possibility, but don't bank on it at the moment. I think just a possible.
I don't like to say never is like never such a never word, isn't it? Never such a finite thing. Anything's possible, really. With stuff like linking to other apps and that, you have to have the cooperation of other developers, and you have to have what's called open APIs, basically, which are just application interfaces that let our app into someone else's app and stuff. But linking data and that, I say, you know, and I'm not really sure. We can be done, but it all depends what permissions we have.
Okay, so we'll have to keep everybody in the loop about that. So we have another question. Saying it looks great. Would there be an option for 2 users on one app, one to input on to review? Parent and teenager is ready to record their own info, but parents need to view also.
So good question that. So what we have got different user types in it. We've got carer, parent and sibling, you know, in it. We haven't looked at like linking apps so a parent can review a teenager's app or anything like that. We haven't thought of that. That's not being mentioned, but we have got the facility where a parent can enter data for a child in it. So no, I haven't really thought of that peer review stuff, to be honest.
And can Irish patients join Radar?
That'll be up to someone who knows about Radar.
No, not really, because
No,
we're restricted to NHS numbers or CAI numbers from Scotland. I don't think so at the moment.
not from Northern Ireland then?
Northern Ireland, yes, but Southern Ireland, no.
Yeah, right.
Because they're not going to be NHS patients.
Right.
Didn't you have a massive data breach a couple of months back as well? Yeah, the massive data breach a couple of months back that they're still not up and running with. We're dealing with a couple of hospitals in Ireland at the minute with another project. And we're just shocked by it all, to be honest with you.
Yeah, I think it's best left for now.
Yeah.
Okay, well, I think that's all the questions. If somebody else has got anything to ask or share? No, okay. Well, I'm very thrilled with the app development so far, Steve. You've done a grand job. And I'm so grateful to all the patients who've given input as well.
Yeah.
And your help, Fiona and Moyne. So I'm looking forward to seeing, when it's going to be completely finished and be able to download it. But we're just going to have to be patient and we'll get there.
There's some processes that we're going to do. We're not going to see. You're not going to be like, from today, you're going to see a final thing in July. There'll be stages and processes for people to have and look at and go, and go through and stuff and give more feedback, because, what we find with a lot of apps is where we start, where we end up are slightly different places as well. But there may be some refining that we do that's needed. As something like what you mentioned before about a third tagging, stuff like that. So it's going to be a journey that you're all going to come along with, and we're going to refine as we go. And it wouldn't be possible without everyone's input, because that's what we like to do. We never come in and go, this is what you're getting, and you like it or you lump it.
No.
Journey and working with people on it. So everyone will get like versions as they go, and we just refine the feedback as we go. All right.
Yes, and after it's been finished and downloaded, we still may have to make amendments afterwards as well. So it'll be evolving.
Yeah, I mean, that's the beauty of doing apps though, because we can, it's like, it's iterative really, so that, once you get it out and it goes on trial, we'll get loads of feedback, and then we can, say which that feedback put in, and then we can just update it, and everyone get an automatic update as well. So just to say, it's cross-platform as well. So it's actually going to be on the iOS and Android too. So, so again, that's big because we got our own bespoke engine that does that. It's not quite with the touch of a button, but essentially, so it's going to be in both platforms.
Thank you, Steve.
Okay.
And just to let everybody know, we'll have future virtual roadshow events, and also some face-to-face roadshow events as well around the country over the next coming months. And I'll keep you all posted on our website and on our Facebook page. And I'll get Miriam to send a memo around to all the renal units as well. Okay, thank you very much, Steve, for your presentation.
Right.
Thank you.
Thanks, everyone. Thank you. Bye-bye.
Bye-bye. Bye. So, I'm going to ask Moyne now to kindly talk to us about what's happening at the laboratories in regards to the research, the Nurture and Nephros research, which possibly some of you're involved in at the moment. And afterwards we'll have a Q&A as well. And if you could kindly put your questions in the chat box for us, please.
Yeah, thanks, Wendy. So I'm going to give you a little bit of an overview on kind of Nurture and the kind of registry work that we're doing in patient cohorts. And I think Gavin's got more detail on the actual research. So are we tag teaming here, Wendy? Is it me and then Gav and then questions?
Yeah, well, it's going to be you. Then we're going to ask people to ask you questions, and then Gavin's going to follow you, and then Martin's going to speak, and then we're going to have a Q&A after Martin's spoken.
Okay, so I'm going to keep mine quite brief then, because it's really just an overview of the Nurture project, and then the meat of it hopefully should be coming with Gav and others, and obviously with the discussion. So can you see my slides?
Yes, yeah. They look good. Yeah.
Just make it into a presentation. There we go. Okay, so clearly I'm going to talk about nephrotic syndrome and the research we're doing into nephrotic syndrome. And the fundamental problem that we have with nephrotic syndrome is that we don't know what causes it. So we tend to define it by the way that we observe it, and we observe things like the response to steroids, or we might observe what it looks like on a biopsy. But none of these are really, they're far from perfect. They don't tell us what's causing the disease and what the different types of disease is. And then they don't crucially point us towards what the right treatment would be. So we tend to use a lot of non-specific treatment and a little bit of guesswork in terms of the best treatment for the best patient. So what we're aiming for is the ideal treatment by stratifying patients into individual groups where their disease is very similar and caused by the same thing. So it's allocation of ideal treatment, treat the right patient and treat them right the first time they get their treatment. And what we've managed to do so far in terms of where the field is that we've managed to stratify patients into patients with a genetic disease, what we call a single gene disorder, and where they've got a mutation in an individual gene that causes their disease, and these can be inherited. So sometimes they're called familial nephrotic syndromes. And then, oops, what's going on here? Then we have what I call circulating factor disease. So this is probably some toxic factor that's released probably by cells of the immune system, so an immune-mediated disease that goes and damages the podocytes, which are the crucial cells in the kidney that are targeted in this disease. And we have some ways and some clues in terms of being able to recognize patients with this type of disease. And then there may be other mechanisms. If we can find ways of identifying those first two groups, then patients who fall outside those groups could have other mechanisms which responds to different treatments. So how do we study this disease? Because it's still a rare disease. So the best way to study diseases is to collect as many patients as you can and study patterns of disease and be able to collect samples from those patients to study the biology of the disease. So we started with the radar registry, and within the radar registry, we started with Nephros, which is the Nephrotic Syndrome study. And this is web-based, and Fiona, who's on the call, has been central in setting this up over 10 years ago now. And the current collection is over 3,500 patients. So it's been fantastically successful in collecting patients from all over the country onto this registry. And some of the benefits are that we can collect both adult and child patients. We can do things like genetic analysis. I've just told you about the genetic subgroup of patients, and we can look at the genetic architecture of these of patients, if you like, and also the degree of or the number of patients across the country who have a genetic disease. And then we can also use patient samples to discover what we call biomarkers. So a bit like having a blood test that tells you what kind of disease that patient might have. So we can look in detail at the blood and the urine and biopsy specimens to see whether we can really tease out what it is that, what patterns we can see that align to different types of disease. So how do we continue this momentum? So we need more patients. That's always the case, and that's always hugely valuable. And then we need to be able to study the patients. So for example, work out what are the signatures in each subtype of nephrotic syndrome using very advanced technologies that can really derive information from individual patient samples, and as many patients as we can, and as much clinical details on those patients as you can. So things like the app that we've just looked at are really critical in being able to get the full clinical picture of the patient that aligns with that molecular signature that we're deriving. And then we can then stratify the patients into distinct groups and work out which is the best treatments, including some of these new treatments that we have potentially available that people have suggested could be useful in nephrotic syndrome, work out which groups will respond to which treatment, and also discover new treatments according to the biology that we are uncovering. So that's where Nurture came from. So Nurture was a development out of the Renal Registry, and this was again a cohort study to build big cohorts of patients, both with nephrotic syndrome and with chronic kidney disease. And so it's a unique collaboration between two different cohort studies, but closely linked. And you can combine the resources, and that was the value, that we could attract funding to combine resources, reduce costs, so that we can deliver both cohorts. And this was very attractive to industry partners, who are crucial in terms of partnership, because they can help us develop those new drugs. Also very, very central to this was Kidney Research UK, and also this sort of principle and the dream, if you like, of establishing the first UK national renal sample repository. So this is a central repository of samples kept in the UK Biobank in Milton Keynes, where all the patient samples are stored. So it's what's called a prospective study. So patients were collected. We had a detailed protocol of how patient samples are dealt with, what information we collect from the patients, and questionnaires. And so, as I say, the biobank includes lots of different types of specimens from each patient. The idea was, because we had good funding from industry, we could recruit rapidly by employing nurses across different centres. And we could also use databases such as the Renal Registry to obtain very comprehensive data on each individual patient. And so we had 14 sites in the UK, and for nephrotic syndrome, the goal is 800 patients, and we're not far off now. We're close to 700. We're collecting biopsies as well, so biopsy samples where they're available, and also follow-up blood samples. So we have a baseline blood sample, blood and urine samples, and follow-up samples for patients who go into relapse and then remission. And follow-up is long-term, so we've said up to 15 years, but we'd clearly like to follow up patients for life, ideally, in order to really get a comprehensive picture of what the disease patterns are. This is just a list of the centres across the UK, and you can see how it covers the whole of the UK. Well, yeah, the whole of the UK, including devolved countries. So the strategy, once you've collected the patients and the samples, is to enhance that collaboration between academia, so the academic and clinical labs, and industry, because industry is key to accelerating drug discovery, which is what we all want, and development of those drugs. So this is a really, really novel and important partnership. And as I say, to achieve it, both industry and us as academics are aligned in identifying correctly stratified patient cohorts to clinically trial new drugs and to discover new biomarkers. And with that in mind, with that goal in mind, we got a very substantial award of money from the Medical Research Council to pursue this. So it demonstrates the success of the whole endeavour. And we had a grant that covers over 4 years. We'll come to an end at the end of this year, and Gavin can talk about some of the exciting research that is coming out of it. And the aim is to utilize the valuable patient data that I've mentioned, all those biosamples, and really try to establish a kind of redefinition of nephrotic syndrome according to discovery of new molecular signatures and markers of disease. And that's through a detailed analysis of all those different samples using the most up-to-date technology. And once you've derived all that data, all that information from patient samples, you need also to have the bioinformatics skills, so computing skills and mathematics skills, to be able to put it all together in a meaningful way. And that's what we've built with the team that with the money from the MRC. And importantly, patients were involved in the MRC project. So we had stratified medicine champions, we called them. So these are designated people, patients and parents from around the country who can represent that patient voice, link the research to the patient community, try and inform the community what's going on, and also help with encouraging participation in the studies, because if patients don't participate, then clearly none of this can happen. And as we're talking about today, Wendy and Polly Mosley originally got this fantastic grant from the Wellcome Trust to develop the patient app that is now coming to fruition. So that's the kind of summary of where we are then. So we've got what we're calling a precision medicine approach, or a stratified medicine approach to nephrotic syndrome, where we're using nurture to collect what's called deep phenotyping. So lots of information about each patient, clinical information, and biochemical and biological information. We're doing genetic typing on the patients, and we're getting biomarkers. We're studying the biomarkers, so studying in detail urine, plasma, biopsy samples, etc., in order to get the right treatments to the right patients. So thank you from the Bristol Renal Group in particular. That's an old photo, but most of the people from Bristol Renal, and lots of people in the study team, including Mariam, who's here, Liz, who's the project manager for Nurture, and people up and down the country, and our university collaborators as well, and the registry, the charities, and Fiona and Rita from the registries who've really helped us get this far. So that's a big thank you to everyone, and happy to take questions, I think, Wendy, at this stage.
Thank you, Moin. I'm always in awe of all the hard work you and the team do, and every time I sit in the meeting, it's very exciting to hear your progress. And yes, thank you very much. Yeah. I don't think we've got any questions at the moment.
It's always great to have your feedback, Wendy, in those meetings, I should say, to have patients.
Oh, thank you. Yeah, thank you. Well, if there is any questions you think of, anybody, then please do email me, and I'll be happy to pass these questions on to Maureen or Gavin, and then reply back to you as soon as possible.
Yeah,
that's lovely. And if people still want to sign up to Nurture, that they still can, can't they?
Yeah, absolutely. You have to.
That's running to the end to December, isn't it? To sign up.
Yeah, you have to be in one of those recruiting centres, because the protocol is very strict in terms of how the blood samples are dealt with. So only those recruiting centres can do that. That can follow the protocol because they've been trained to do it.
Yeah.
But as I say, there are centres up and down the country that cover most of the UK.
Yeah, that's great. And if anybody wants to sign up, you just ask your consultant, then you'll be given, a nurse will go through forms with you for your consent to sign up.
Yeah, if you want to see what the centres are, because I only flipped through it on my slides quickly, is if you go to nurturebiobank.org, so nurturebiobank, all one word, .org, that there's lots of information there, including all the centres.
That's lovely, thank you. And just a couple of comments in the chat box. Thank you so much from all of us. Thank you for all you do. It's much appreciated. Okay, so we're now going to hear from Professor Gavin Welsh in regards to the important research which is happening. You can tell us some exciting news, hopefully. What's been happening? What's been going on lately in the labs?
Thank you, Doug. So can you see my screen?
Yeah, that's lovely. Yes.
Okay, brilliant. So I just want to follow on from Moin and explain some of the things we've been doing in the lab, especially what we do with all these samples that you are collected from Nephros and from Nurture, and why these samples are so valuable to us. So to go back to the beginning, what are we interested in the lab? Well, our lab work bases around the kidney, which is the world's most sophisticated filter. And we want to understand how this filter breaks down in nephrotic syndrome, and can we reverse it, or can we get new treatments which are desperately needed for this disease? So what we're interested in is, this is the kidney on the left. I don't know whether you can see my pointer, but we're interested in these structures called glomeruli on the right, which are the filtering unit of the kidney. So you've got about 1 million of these in your kidneys, and they filter the blood to produce the urine. And when that breaks down, you get kidney disease. So these glomeruli are a tri-layer structure, which forms a sort of sieve. So it sieves the blood, gets rid of impurities which come out of the urine, but importantly doesn't let any proteins go into the urine. So we're interested in this whole complex. And Bristol Renal, which is a big group of about 30 to 40 scientists, are interested in the whole structure of this kidney, and we all look at various different parts of it to try and understand how it breaks down in kidney disease. So this is a further part of it. This is the glomerulus again, and showing on the right-hand side, you can see these beautiful, very tight structures, which is the filtering unit. And this is what we're interested in the lab. And if you look at the structure with all, you can see these round balls of capillaries. That's something important to keep in your mind for what I'm going to talk about in a bit. So how does it work? Well, the blood enters the glomerulus, it travels through the glomeruli, and in a healthy person, it's filtered, the blood exits and it's been purified, and all the waste flows out of the glomeruli through the tubules and becomes urine. How does the filter work? Well, the glomerulus is a very highly selective filtration barrier. It filters the blood to produce urine. It's made up of 3 layers: endothelial cells, which are on the blood side. You've got this thing called the glomerulus membrane, and then you've got Moyen's favorite cells called the podocytes. And this 3-layer structure is what is required for the filtration. And if damage to any of these parts of the filtration barrier causes disease. So we're interested in this whole barrier as a whole, and how does it break down in not just nephrotic syndrome, but other kidney diseases. So this is one of my favorite pictures, and this shows Moyen's favorite cell, the podocyte. And you can see the podocyte's like an octopus, we call it, because it's got this big cell body, you can see in red, and then it's got all these foot processes which come out, and they join together, and that forms the part of the filtration barrier. Again, remember this structure because I'm going to come onto it in a minute. So here's how it works. You've got this 3-layer structure. 180 litres of water pass through this structure, but in a healthy person, no protein goes through. And that's important because leaking protein into the urine is a cardinal sign of kidney disease. So what happens when this amazing filter breaks down? Well, you start to lose essential plasma proteins like circulating antibodies, clotting factors, albumin. The glomeruli start to be lost, and that loss of the glomeruli leads to chronic kidney disease and ultimately end-stage renal failure. So can we understand how this filter breaks down? So as I just repeating it, this is, you lose these essential circulating proteins, but the mechanism how this filter breaks down is still unknown. And it's a huge disease burden and complex outcomes post-transplantation. So we really want to see, can we find new treatments for the disease, which are really necessary. So here you can see the nice normal glomerulus on the left, and this breaks down, you get scarring in the damaged kidney. And so we want to see if we can model this whole thing in a lab, because at the moment, current models to look at kidney disease in the lab, again, aren't very useful. So our aim is, as Moyen said, we've got patients with nephrotic syndrome who've joined Nephros and Nurture, and we're now taking samples from them, but very highly collected samples, very specially collected. They've all got to be collected to industry standard, which means we've got this huge biobank of samples sitting in Milton Keynes that we've got access to, that we can now use in the lab to do experiments on. So if you see on the left, this is a normal glomerulus. This is the normal podocyte structure. You can see the lovely foot processes. And in nephrotic syndrome, this breaks down. So you lose all these foot processes. The podocytes become effaced. And that means they sort of flatten, they lose their structure, and they stop filtering properly. So the question we asked is, we've got the cells in the glomerulus in the lab. So Moyne, about 20 years ago now, managed to make these cells from the lab, make podocytes, human podocytes from discarded kidneys, so we can grow them in the lab. We also grow the endothelial cells, the other side of the barrier. And these cells have been used by all the pharmaceutical companies around the world. But this is what we call 2D. They grow separately in a dish in the lab, a plastic dish. And actually, it hasn't taken drug discovery forward because these cells like to talk to each other. They haven't got this beautiful structure on the left. So the models that they use for drug discovery are very poor at the moment. So we thought, well, as we've got the cells in the lab, can we put them together and get a better model of the glomerulus, which is better used for pharmaceutical companies and for us to study nephrotic syndrome? And Jack Tuffin in the lab spent a lot of time developing techniques using special magnets to put these cells together. And what he found was, if he treated these cells together and forced them together with magnets, he can form a structure like a glomerulus in the lab. These cells have got amazing ability to self-organize, so they know where to go. So actually, if you put them together, they don't just form this useless mixture. The podocytes know they should go on the outside. The endothelial cells know they should go on the inside. So we've actually got a structure now that looks like a glomerulus. So here, if you remember back to what I said, here's our new glomerular structure that we can grow reproducibly in the lab. So this work was part funded by NEST. It's just been published, and this is a really useful tool for us now to study these structures in the lab. So Moyne mentioned Nurture and mentioned the fact that we got all these fantastic samples really carefully collected. So what we can now do is use these structures and all the plasmas we're getting from all the patients in Nurture and do experiments in the lab to try and understand what's going wrong in nephrotic syndrome, in the case of nephrotic syndrome, and also to test we've got some ideas of how the damage to the podocyte is caused, so we can use these models in the lab to see if we can test drugs to block these deleterious pathways. Also, we're working with various companies who have now got what we call spheroids, and they're using them in the lab for high throughput screening of drug compounds. And again, they're finding these structures much more useful and more representative of what happens in a human condition. So this is very exciting for us. We're working with Evotec and UCB to use our structures, to use these new spheroid structures for drug discovery. So I think there's a lot of mileage in this that hopefully we can, working with the companies through the Nurture samples, we can find better treatments for nephrotic syndrome patients. The other thing, excitingly, Moyne and I have been involved with is a new gene therapy way of treating kidney disease. So a lot of nephrotic syndrome patients, as Moyne said, have a genetic form of disease, i.e. there's something wrong with the DNA sequence. And we were wondering whether we could take the cells have a defective gene, which causes the podocytes to be damaged. And we were wondering whether we can introduce the healthy gene to restore the cellular function of the podocytes. So can we treat kidney patients with gene therapy tools like have been used in a lot of other tissues. And excitingly, gene therapy is a huge increase in gene therapy tools coming out. And we wondered whether we can do it for the podocyte to treat nephrotic syndrome. So this was started by Dr. Wenyi Ding, who developed viruses that can directly target the podocyte within the kidneys and hopefully give them functional copies of the mutated gene. So can we treat nephrotic syndrome patients with this new technology? So what we're using is this what's called adeno-associated virus. Most people have adeno-associated virus within their bodies anyway. It's a very safe virus. It's being used for gene therapy throughout the world now, but not in kidney disease. So we're working to design new ways of introducing this AAV to the body to directly target the cells of the kidney to give them functional copies of the mutated genes. And hopefully we can cure disease in this way. So through Nurture and through Nephros, we and other people around the world have identified about 70 to 80 genes now which are mutated. The mutations give you nephrotic syndrome. So there's a huge bank of genes that we could possibly target to treat various forms of nephrotic syndrome. So we think this is a really exciting new way of trying to cure kidney disease. Excitingly, Bristol, our lab has secured a £45 million investment from the Wellcome Trust, Syncona, to set up a new gene therapy company, which is now established in just near King's Cross in London. So we're now taking this forward with this investment, and we're hoping that we can develop new cures for kidney disease within the next few years. So this is really exciting, and Moyne and I are very proud that we've managed to get this. But this is built on work that Nest has funded over the last few years of work we've been doing in the lab. So this is a really exciting way of doing things. So hopefully that's just a flavour of what we're trying to do in the lab. Hopefully you can see there's some exciting things going through. As I said, treatment options for the glomerular diseases like nephrotic syndrome at the moment are non-specific. They've not altered in decades. We're trying in the lab to understand the basic biology of the glomerular cells so we can think about how new ways of treating disease. And we hope these advances will lead to novel targeted therapies, hopefully within the next few years, so that we can start treating these devastating diseases. Thanks for listening.
Wow, thank you very much. A wonderful presentation there, Gavin. Yeah, very impressive, all the work you're doing at the lab. Yeah, thank you so much to you and the team. And we will look forward to hearing more in the next coming months. And hopefully you can pass that on at face-to-face meetings we're going to have, soon. So that's fantastic. Thank you so much.
Yeah, thanks, Gavin. It was very clear and very understandable the way you presented it for certainly someone that doesn't know 100% about it, and it was really good. So well done. Thank you very much.
Thank you.
Thank you. Okay, that's lovely. Thank you. So our next guest speaker is Dr. Martin.
Sorry, Wendy, I think there's a question in the app.
Oh, is there a question? I do apologise.
COVID vaccine, so I'll pass that on to Moyne as a clinician. I'm just
okay.
Cell biologist.
Okay, thank you. Okay, so Dr. Martin Christian is going to be speaking next, and he's going to give us the result of the Pregnus 2 study. He's with us. Yes, he is. Yes.
Hi, Wendy.
Hi, Martin. Thank you so much for making time tonight to be with us.
Thank you very much for the invite to speak. Right, let me try and do this screen share. This flitting between Teams and Zoom, isn't it? That you kind of right. take that now, and then so I'm probably going to look away from my screen, a double screen here with a bigger one that's not got the camera on it. So excuse me if I don't look looking at you. But thank you very much for the opportunity to present this. I think many of you will already know about this study, but just by way of background, what we know about relapses in nephrotic syndrome, that many of them are triggered by infection, and many children who have nephrotic syndrome relapses, not only the relapses triggered by infection, but when the children get infection, they're likely to trigger relapses. And studies from around the world have suggested that giving low-dose prednisolone, low-dose steroids, every day for about a week may trigger a relapse. Now, this is a very busy slide. I'm not really going to talk all the way through it, but the top line suggests that the research is important and significant, and the p-values are for probability. So generally, if a p-value is less than 0.05 or even better, less than 0.01, that means there's a...
Martin,
yes.
Your slides aren't moving forward, I don't think.
Okay.
I'm still on your first.
Oh, thank you. Okay, I'm not quite sure how this...
you put it on slideshow, you should just be able to click through it.
Yeah, okay.
There you go.
Tell me if it does it again, because I thought I did this before mine, but there's something funny that happens here. So if a p-value is less than 0.01, what that really means is that the chances of that result being occurring by chance is less than 1%, and that so you can see that the 3 of the 4 studies had that probability, and the other one was less than 5%, which is significant. But there were quite a lot of problems in the size and the fact that they were restricted to children only taking prednisolone for most of them. Some issues to do with the way the studies were designed. They weren't blinded, so people knew what treatment they were having, and that might influence the results. But perhaps one of the most important things was the countries that these studies took place in were all tropical countries where the diseases are different, and we weren't sure whether the viruses that we get in this country triggering relapses would respond to steroids in the same way as infections in these hotter countries. And in fact, when Cochrane, which is an international body that assimilates evidence from different research, looked at this particular question, and it's a question they've been looking at over many years in terms of steroids in nephrotic syndrome. They said that it would be unlikely that doctors would use this regimen without further evidence to confirm whether it's helpful and whether it's safe to do so. So really, that was why Nick Webb, who was my predecessor as the chief investigator for this study, set up this study. And to get around the method problems, it was set up as a double-blind, placebo-controlled study. So neither the research teams locally nor the patients and their families knew what treatment they were going to be receiving. And that meant that when they commented on steroid side effects, particularly subjective ones like behavior, they could do it without being influenced by knowing what the treatment was that the child was getting. And it was a very large study. It took place in 122 paediatric centres around the UK. 91 of them recruited patients to it, and there was 365 children. So that makes it the largest interventional study in childhood nephrotic syndrome that's ever taken place. The study drugs, that's what IMP stands for, were either 5 milligrams of prednisolone tablets or a placebo or a dummy tablet that was designed to look exactly the same. And the way the study set up is that these pots of these tablets posted out to families once they recruited. and they were given instructions about how many tablets to take at the time of the relapse. But this was the schema for it. These are the children we recruited were children who had had 2 or more relapses 12 months before the study. They were randomized by a computer, which assigned them to either receive in the active treatment arm, that is prednisolone tablets, or the control arm, getting the dummy tablets. And as soon as they had symptoms of a cough, of an upper respiratory tract infection, they had a fridge magnet to remind them exactly what the definition of that was, then they could start taking the prednisolone tablets for 6 days. Now, this is the first time I've had a chance to use this photo in a presentation, and I'm really glad to be able to do it, because when I took over the study, I asked about a patient phone, and this was set up in order that if there were any queries from families about taking prednisolone at the time of an upper respiratory tract infection, if their local teams couldn't answer it, particularly if it occurred out of hours, there were calls. There was a national number, and this was routed to this particular phone. This photo was sent to me by Nick Webb's colleague when I asked what had happened to the hot phone. And
Martin, I'm still not seeing beyond slide 3.
Oh,
I don't know if anyone else can.
Okay. Sorry,
Shame to miss your great photos.
Yeah, no. Let me go back. Let me try sharing again here. I think this is something to do with the way you share, isn't it? And whether you share screen or whether you share.
Yeah, you can share a desktop as well, can't you?
All right. I think if I share a screen, I think that might make it different. Let me try it again. Okay, so do you want to just stay on mine and just tell me if that's advanced this time?
Yeah, that's working.
Great.
Yeah.
Okay. Right. Thanks for that. So sorry. This is my photo I'm trying to share with you. That Nick Plant, who is Nick Webb's colleague in Manchester, as I took over and asked about whether this hotline phone was still in existence, he sent me this funny photo, just showing that he's been plugged in their office right the way through the trial with a big note telling people not to switch it off. So 365 children took part in the study, but the final analysis population didn't include that many, and that was mainly because a lot of children, more than we were expecting, went 12 months of the trial without having had an upper respiratory tract infection. Some people withdrew, but had an upper respiratory tract infection before they withdrew, and so we were able to include those data. So we ended up with a population of 271 that we were able to use for the analysis, and that was 134 who took the prednisolone and 137 who had the placebo. And this nice infographic that was produced by one of the papers that we published for it just helps to show all of this through. So that population of 271 children were about twice as many boys as girls. That's with the numbers that get nephrotic syndrome. Their average age was 7.6, and the study took place in 91 weeks, as we said, and they were 271 children were analysed and randomised, and 131 had prednisolone, 137 had placebo. And the primary outcome, what we were most interested in, was the number of first upper respiratory tract infection associated relapses in that 12 months study. So first of all, just to look at how the different populations broke down, and they were pretty balanced for their age, the age of the diagnosis, the males and the females between both groups were balanced, and in terms of ethnicities as well, a fairly good balance of the ethnicities between the two groups. We split the groups into 4 subgroups depending on the type of treatment at the start of the study. So either they were children who weren't on any long-term treatment, they may have been on long-term treatment with just prednisolone. They could have had another immunosuppression like tacrolimus or lubamisol along with prednisolone, or they might just have had this other immunosuppressant, tacrolimus or lubamisol by itself. And the headline news is that we found no difference in the number of these upper respiratory tract infection associated relapses between the group that took prednisolone and the group that took placebo. Percentages are very similar, and when we do the statistics, it shows that there's no difference between them. If we looked in terms of the subgroups according to the treatment that they were on at the start of the study, again, there were no differences between the two groups. When we looked at the secondary outcome and we measured other outcomes as well, like changes in the background treatment, the overall dose of prednisolone, adverse effects of prednisolone or adverse effects generally, and behavior scores like we used for the PredNOS study as well, and quality of life measures, no difference in either arm. The only one thing that was of interest, and this is not a significant result, but it was a little bit less, it showed more of a difference than the overall study, is when we looked, and that's what the background to this analysis is, that we were surprised by the result, given that the 4 studies, they were smaller, they weren't such good studies from a methodological point of view, but they did convincingly show that the prednisolone made a difference. So when we looked at just the children who were of South Asian ethnicity, and this was according to the self-reported ethnicity, and so in that were children whose family from Pakistan or from India or Bangladesh or Sri Lanka, or had mixed ethnicity with one of those as part of it. And we compared them to the other children, 213 children of other ethnicities. There was a bit of a difference, as I've said, but it didn't reach significance. So the p-value, remember I said before, less than at least 0.05, and that was 0.09. So conclusions then, as I said already, this has been the largest interventional study. That's what CTIM means. So an interventional study means giving a drug, trialing a drug in a condition. And it was a study that was designed robustly to give good evidence at the end of it. And we showed that it doesn't make any difference whether you give 6 days of oral prednisolone to try and prevent a relapse caused by an upper respiratory tract infection. Just that hint of a difference, though, in the children who are of South Asian ethnicity. And we've got some samples that were collected as part of the study that we would like to look at in a bit more detail, and look at perhaps some gene profiling to see whether we can detect any differences in those that responded, those that didn't, that might help us understand that a little bit more. We published the study in its journal, JAMA Pediatrics, at the end of December, and it's an open access paper. If you're interested, you can go on and read the whole paper. And the study was funded by the National Institute of Health Research, and this is the publication, a more detailed document of what went on in the study, again, open access. So finally, just the acknowledgements really for, because I came in later on in the study, and the most important person that I need to acknowledge here is Nick Webb, who set up the study and was the chief investigator for the first part of it. All of my co-authors are listed there, who are paediatric nephrologists from different centres, as well as the central team at Birmingham Children's Hospital. Study funded by NIHR, as I've said, but of course, most importantly, the children who took part and their parents who were allowed to take part. And I suspect that some of you listening belong to that. So thank you very much for agreeing to be part of this study. Research networks, the local research teams were fantastic. Wendy and other members of NAST were also brilliant in terms of helping to get the protocol right and steer the subject. So I think that's all I have to say. Thank you again for the time. Happy to answer any questions.
That's great, Martin. Thank you very much for sharing those results. And thank you very much for completing the study, because I know it's a huge amount of work on top of your clinical work as well. So thank you to you and lots of other researchers who undertake these studies to ask these important, to get the answers to these important questions. I don't think we've got any questions at the moment for you.
Wendy, there was a question whether Moyne and Martin could answer.
Yes, Martin, Moyne's replied to Tina's question about the reports about relapse occurring after vaccination. And I know your team is actually looking into this at the moment, aren't you?
Yeah, I mean, if I can take the opportunity, sorry to slightly deflect from Martin's excellent work, but we'll come back to it. But just that it is interesting, and we sent the questionnaire via your Facebook group. Marianne's posted the link there, and we got 41 responses so far, and of those responses we've seen, I can't remember what it is, Marianne, about 7 or 8 patients who definitely relapsed after vaccinations. Clearly there's going to be an element of reporting bias, so we don't know what the overall risk of relapse after vaccination is, but there's at least a small risk. So it would be really important if you can encourage people to get more responses, because if we can up those numbers, we would much better.
I need to put that link up again on the Facebook page. So our Facebook page goes rather fast at times, so I need to keep I think the past week I actually haven't put it up, I must confess. So I'll put it up. I keep putting it up to get more responses.
Just put it up once an hour on the hour would just be about right.
Okay. I'll do that then. Okay.
And then we'll come back to Martin. I mean, I should, for Martin's work, I should emphasize how hard it is and how amazing it is to do that huge study in nephrotic syndrome patients. We've said how rare the disease is and how difficult it is even to get a registry, patients on a registry, but to do a trial in that number of patients is just astounding work.
Yeah, it's a huge undertaking. Yeah.
I mean, I can't take credit for that. I think it emphasizes, though, that it's been a really strong research infrastructure, and we're trying to set up another study to look at relapses, which we're struggling to get funded at the moment, but still trying. But the research infrastructure is there, and it would be really good to continue to make use of the good relationships that we've built up, because I think we can continue to answer these sorts of questions, and starting to bring the clinical trials and the nurture work together offers a really powerful tool to start to begin to answer some of these big questions we've got.
Yeah, and it does give us an opportunity. If we come up with some key questions through nurture, for example, then with Martin's trial network, actually would be in a very unique position to go and start to answer some of those questions and trial new therapies.
That would be fantastic. Yeah, we look forward to that very much.
Wendy, can you ask them to explain my query I've just posted on a chat? Or would you like me to read it out?
I think you better read it out, Fiona, because I can't
sorry.
find it.
Right, I thought it wasn't coming through. One of the reasons we were asked to set up the app was to be able to collect samples during relapses, and I would really like to know what you're going to do with those, both teams of researchers, and can you explain what it will add to your research?
Yeah, so shall I start answering that, Fiona? That's a really question and a really important part of our research. So these samples during relapse are really valuable and also really difficult to get. So we've tried quite hard, and Fiona's helped us with this as well. And the reason that it's so valuable is when you're relapsing, that's when your disease is most active. And so if you're looking for what is triggering disease and what is driving the disease, what you want is samples from the patient when that disease is most active, because that's likely to show the signs of, say in a blood sample, the signs of what it is that triggered the disease, and which cells in particular of the immune system may be the most important cells in triggering that disease. And if you know that, then there are lots and lots of drugs out there that target very specific cells of the immune system. We might get real clues into using even existing drugs, let alone needing to develop new drugs. So there are lots of existing drugs that could be tried. So that's the main reason, and the kind of things that we do with those samples include something for example called transcriptomics. And just to explain what that means very sort of simply, it's if in every cell in a blood sample, each of those cells has a specific profile of what's called RNA. So it's the genes that are switched on in that cell out of the whole of your genome, or every gene in that cell. Every cell contains every gene that an individual possesses, and what makes that cell unique is which of those genes are switched on or off. So transcriptomics is a very powerful way of seeing which genes are switched on and off in individual cells. So we can take a blood sample, we can look at the individual cell level, which genes are switched on and off. And if you can find a pattern of genes that are switched on from a particular cell type from a patient in relapse across quite several different patients, then you can start to work out which are the important cells and which are the important genes that are switched on during that most active part of the disease. So I hope that's not sort of making it too complicated, but that's one of the key reasons that those samples are so valuable.
Would the cellular pictures look different in relapse?
The cellular pictures?
Yeah, the pictures that Gavin's just shown us, would they look different?
Yeah, so that's another way of analysing those samples. So what you do is you take the sample, you make an assumption that in that sample is some toxic factor that damages kidney cells, the podocytes. And so you put the sample, you dilute it a bit, you put it onto the podocytes in the dish, or that's the sphere that Gav showed, and you see what happens to it effectively. You have a readout of what happens to that cell or that glomerulus, and that again will build up a pattern that you could interrogate to see what it is that's driving it.
Okay, that's really helpful. Thank you.
We've got a couple of more questions in the chat box, Moin. Would the app be useful for a relapse study?
Yeah, I think so. That's sort of building on what we just said. So if the app sort of signposted people who are relapsing, then maybe we could approach those patients, actively during their relapse and say, please come to hospital and give us a sample.
Great, thank you. Can you say any more about some of the new treatments you mentioned?
So there are lots of possible treatments, and the ones that are most actively being tested at the moment, and maybe Martin could add into this, are things like rituximab, which most people know about. So what are called biologics, so treatments that target specific cells of the immune system and only take out, a bit like a sort of targeted cruise missile, only take out a single cell type amongst many cells that are circulating in the immune system. So some of those, and there are lots of different types of biologics around now, and rituximab's the one we use, but there are many other potential ones that could be trialed, but we just don't have the knowledge yet to know which ones to try. So those are some of the most exciting ones. I don't know whether, Martin, you want to comment as well on other treatments that are potentially, that there's different sort of steroid regimes that we could trial as well, aren't there?
Yeah, I don't think there's anything more to add really at the moment, Moin. I think there's a lot that we don't know about the basic treatments that we use, about which ones are better necessarily than others, or and things like rituximab, which has now become commonly used. Should we be waiting until we've exhausted all the other treatments, or should we be using it earlier on?
Yeah, we've had an interest in a subset of cells called Th17 cells, which we've published some work on from the lab studies, from using patient samples, etc. And that looks like a promising one, where there are drugs that can specifically target those type of cells for other diseases. They're used in diseases such as psoriasis, for example. So we're quite interested in trialing some of those, but you need to build up the evidence, and that's where all these disease samples come in really to the fore and become really important.
Wonderful. Thank you. Thank you very much. I don't think there's any more questions. If anybody thinks of further questions, then please email me. It's wendy@nstrust.co.uk. And I'll pass those questions on to whichever speaker you wish the question to be directed to, and I'll get back to you as soon as possible. So thank you very much to all the speakers that come on this evening to speak, and thank you for your lovely presentations. And thank you very much to Miriam being my technical support and hosting this meeting as well. And thank you everybody for joining tonight, and I'll keep you informed about the app, and we'll have further meetings, and I'll give you all the dates on Facebook and on the website. Thank you, everybody.
Thanks, Wendy. Well done.
Thanks, Wendy.
Thanks, really.
Thank you. Bye.
You all have wonderful work. Thank you. Bye-bye.
Stride.
Take care.
Doing so.
Bye-bye.
Bye all.
Thank you. Bye.
Bye. Bye. Bye.